Okay. So we were talking about methods of patterning cells, proteins, and surface properties. And last class we covered self-assembled monolayers. Today we're going to talk about biomolecules and how do you pattern biomolecules and how do you pattern cells on substrates. So this would be relevant to your project, surely. I wanted to add this slide. I didn't have this earlier because I want to give you all an idea of why we would want to modify a surface to begin with. A few examples you've seen already, and a few examples may seem new to you. One example is tuning the surface properties. All right, Jeremy, you brought this example of Rain-X, right, last time. If you treat a surface with you can treat a surface to change it from hydrophilic to hydrophobic. Glass is hydrophilic by nature, so the water likes to adhere to the glass, and you could make it. Let me zoom in on some of these things here. There we go. By treating the surface of the glass, you can make it hydrophobic, where the water would beat up. Right. We looked at some examples of hydrophobicity, super hydrophobicity, and so on last time. Sometimes something simple as a surface treatment. We talked about super hydrophobic coatings. If you take a hydrophobic material and then you texture it with, you put little ridges on it, it'll become very hydrophobic. And this was the example of that ketchup bottle. I'll upload a new version of the notes today, so if you want to check out this link, there's a company called Liquid Glide that's making these very slippery surfaces for, I don't know if they're doing it for ketchup bottles anymore, but this was the initial example that they showed. It's pretty cool. No ketchup on the bottle of this. Isn't that great? Now, Liquid Glide is actually doing something interesting. They have surfaces that are impregnated with a liquid. So it's a surface that actually has liquid-like properties. It's very slippery. So these are examples where you just want to change the overall surface properties of the device. Another example is where you would like to, for example, capture cells on the device. Now, in this case, what you would do, we're going to talk about a few techniques today, but you could have molecules attached to the walls that capture cells. Okay, for example, right now, a lot of you know about a little bit how cancer works. Cancer cells, you might have a tumor in one area of the body, and that tumor some of the cancer cells invade into the blood vessels. And once a cancer cell gets into the blood vessels, it goes through the blood and it deposits itself in a different part of the body. This is referred to as metastasis. And this is how tumors spread. So one of the ways that over the last 6 or 7 years, there's been a lot of interest in diagnosing cancer by looking at circulating tumor cells within the bloodstream. And this is a famous paper that was by Sunita Nagrat. She's actually a faculty at University of Michigan, just down the road, and Mehmet Toner, who's like a pioneer in microfluidics. He's like one of the very well-known people in the field. They did a circulating tumor cell capture device where they had a series of microfluidic channels running in parallel, and they'd flow blood through this device. Okay, and you can imagine that blood is made up of a whole bunch of it consists of a liquid part. It also consists of a lot of blood cells in them. The challenge with capturing circulating tumor cells is that out of, let's say, 1 billion cells, there's only one circulating tumor cell. Okay, so you want a structure that will pass through most of the cells, but will only capture the cell type that you're interested in. So what they did is they coated, they had this microfluidic channel that consisted of a series of posts. The posts were there, as you saw in your homework assignment, to increase the surface area. It increases the probability that the cell will collide with the wall. Okay, and the relevant thing here is that they also attached antibodies. They functionalized the surface so that the surface is coated with antibodies. Antibodies stick to cells. Okay, for example, here they had a certain antibody that was specific to circulating tumor cells, that only would capture circulating tumor cells. So other cells would go up and bump up against the walls and just continue on. Circulating tumor cells would bump into it and stick. By doing this, they're actually able to not only capture the circulating tumor cells, but also study it in situ using microscopy inside this microfluidic channel. Okay, so this is an example where the surface is functionalized to actually capture something. Other examples of this is, one example has to do with cell and tissue culture. We saw this example in the first lecture when I was talking about organs on chip. This is a relatively recent Nature publication. They have two microfluidic channels separated by a membrane. In the top channel, they flow in one cell type, in epithelial cells, and on the bottom channel, they flow in endothelium cells. The membrane is functionalized so that the cells can attach to it. Okay, so we're intentionally trying to attach cells to the surface so that they will grow properly inside the microfluidic device. This particular device was meant to mimic the structure in the lungs, where you have a two-layer system consisting of epithelium and endothelium. This side is exposed to air, the alveoli in your lungs, and this other side is exposed to the bloodstream. So a lot of times in microfluidic devices, we want to create structures, like biomimetic structures, that are similar to what's in the body, because cells behave differently when they're in vivo. Okay, if you try to recreate the environment where what it's like in the body, the cells will actually behave differently than if you just plate them out on a culture, on a petri dish, what biologists have been doing for so many years. Okay. In vivo behavior versus in vitro behavior. In vitro is in a dish, in vivo is in the body. Okay, so you want to functionalize surfaces so that certain cell types can attach. This is another example by Sangeeta Bhatia's group. They're doing liver tissue engineering. In liver, there's two types of cells. The liver cells are called hepatocytes, and there's two cell types. And it's been shown that if you grow liver cells out, ex vivo, like just on a dish, just a single cell type, those cells do not function as they do inside the body. There's actually some intimate communication that happens between the two different cell types that causes the livers to secrete certain enzymes and to behave as a liver tissue would in the body. So her group did a lot of nice work on co-culture. Now, the problem with patterning here is that you want to pattern one cell type into certain regions. So you can see that the red dots here is one cell type. And then you want to pattern a second cell type into other regions. Okay, so their goal here was they were trying to create structures that mimicked the certain structures in the liver, where you have two cell types in close communication with one another. So in this type of situation, you'd want to pattern the surface to attach a single cell type, for example, the red cells, and then you'd want to functionalize other areas to attach another cell type. And it is possible to do these types of things. A device that became technology that became very popular in the 90s. It's not quite as popular now, but it's been supplanted in some ways by DNA sequencers, is these microarray based devices. Now, in these devices, you have a small surface like this, and in that surface, there's a whole bunch of different dots on it. Okay, those dots that you see here, this is where DNA attaches to it. This is a DNA analysis device. We haven't talked that much into the details of how DNA analysis works, so I'm just going to say that that each one of these different dots attaches a different type of DNA molecule, a different DNA sequence. The DNA microarray may contain 25,000. For example, this product contains 25,000 dots, 25,000 different oligonucleotides, 25,000 different DNA sequences. When you analyze a cell, you can tear up the cell, collect the DNA, and then what will happen is that the cell's DNA will hybridize, or it will attach to certain spots in this DNA microarray. Okay, and depending on which and when they do attach, that causes the DNA microarray to light up in those spots because the DNA is fluorescently labeled. What this allows you to do, it allows you to actually measure the amount of mRNA expression. It's hard to explain this without going into the details of the cell biology, but basically the type of behavior of the cell, one way you can characterize the behavior of a cell is by looking at how much of a certain type of RNA, messenger RNA, is being expressed. That messenger RNA will bind to these DNA, to these microarrays, and you can monitor whether a cell is expressing a lot of this gene or a cell is expressing a lot of this other gene. Muscle cells will express a lot of certain enzymes, and a liver cell will express a different set of enzymes. Okay. Like I said, I'm not going to go too much into detail right now. I don't want to get caught up in the details of it. How it's relevant here is that you actually have to pattern the surface in order to capture DNA molecules at different spots in the array. Okay. So the last few examples here were about how we'd want to modify a surface to capture biomolecules. And that's what this chapter is mostly about. But in some cases, you also want to modify a surface to prevent molecules from absorbing. Right? I mentioned to you last time that the coronary stents, these are implantable devices. This is implanted using a procedure called angioplasty, where the surgeon will take a catheter, put it into your blood vessel, and then this balloon, you use a balloon to actually expand the stent so that the stent forms this three-dimensional, like tubular shape. Now, does anyone know what stents are used for? Maybe some of you.
If you have a pipe clogged into your blood vessels.
Exactly. Exactly. If you have a blockage in your artery, or if you have fat deposits in your artery, and that's causing your arteries to be thinner, constricted, then what you do is you can take a stent to actually, first thing you do is you do angioplasty. You blow up a balloon inside the artery. And by blowing up the balloon, you're putting pressure on the walls. You're causing it to expand. So you're opening up that blockage. And then you put in a stent, this basically a metal structure here, to make sure that it stays open. Okay. So the stent is made up of a metal that's biocompatible. However, in order to ensure that cells don't start to stick to it, because it is a foreign substance, right? Your body's going to think, oh, this is foreign. Okay, the immune cells are going to go and attach to it, or other cells are going to attach and form biofilms on it, and so on. In order to avoid that, the stents have to have biocompatible coatings. So these types of coatings will resist the absorption of proteins. They'll resist the absorption of cells. Okay. It's the opposite of what we're doing here. Another example is a neural microelectrode arrays. I mentioned this in class last time. I thought it'd be nice to show you a picture. These are what the arrays look like. It's pretty interesting. They're basically an array of like very long and sharp needles. This structure, believe it or not, is actually put into the brain. These needles are very, very small. You can see that this dimension here is just 3 millimeters. Each one of these spikes is actually an electrode that records from the brain. These electrodes actually record from the tip of these needles here. So you can actually read brain activity because the neurons work, they give electrical impulses. You can read that electrical activity directly by using these electrode arrays. The reason they have an array of electrodes rather than just one is because it allows you to do mapping of the brain. This neuron fired at this time. This neuron fired at this time. So you're detecting from more neurons at once. And you may or may not know this, but they've actually developed maps of which part of your brain is firing when you move your finger, which part of your brain is firing when you move your left toe. Again, you can not only sense it, but you can actually control it too. There have been examples of where they use they can stimulate certain areas of your brain, and that will cause certain body parts to move.
What's the varying depth of the needles? Is that for different interactions? So like it has looks like a...
Yeah, actually, so this is a relatively newer picture. The first generation of electrode arrays, they were all the same height, but that means they would all be recording from the same layer of the brain. It turns out that different layers of the brain are responsible for different activity. So that's why they have these arrays that have varying heights. So they can simultaneously record from different depth layers. So these are used a lot by neurophysiologists right now. So these are some of the reasons we'd want to modify a surface. This is, electrode arrays are another thing where you can get an immune response. If you have the proper coating on them, you can prevent some of those responses.
Would you use similar things like that for like the microsyringes and stuff? I read an article about microsyringes. That's kind of cool.
Oh, drug delivery needles. Yeah, any implantable device, you have to think about biocompatibility. Actually, yeah, the microneedles that you mentioned, they're made using a similar idea here. You still have these needles, but they have a hollow tip, and they're hollow needles so that you can inject a drug into them.
And these are not. These are just like a.
These are actually solid. Yeah, they're solid and they're conductive, but most of it's coated except just the tip. The tip is not coated, so that part is the recording electrode, the recording site. Yeah, so you want to coat most of the needle with some type of biocompatible coating, and just leave the tip of it exposed because you need to have the metal part of it exposed to detect the electrical signals. Okay, so last time we talked about self-assembled monolayers, all these things, so I'm not going to go into that. So let's get into biomolecules. So micropatterning proteins. So similar to self-assembled monolayers, there's a few different ways we can pattern proteins, and so we'll go over some of the methods. Photolithography, microstamping, microfluidic patterning, and self-assembly. You'll see a lot of these techniques they're just in some ways they make sense. They're related to things that we talked about in the previous module and in last class. So if you want to pattern proteins and cells using photolithography, you can use this type of process here. We talked about self-assembled monolayers last time. There's two types of self-assembled monolayers, the alkyl silane and the aminosilane. Do you guys remember which one can be good for attaching cell types? This example will show it. Alkyl silane is something like that has a hydrophobic group attached to the end, CH4. And an aminosilane is something that has an amine group, an NH group attached at the end of it. So this is going to show how you can take advantage of those differences. So the first thing you do here is just deposit photoresist, pattern the photoresist, and in some areas you just deposit an alkyl silane self-assembled monolayer. That's shown by these circles here. All right, this is terminated by a CH4 group. And then what you do is remove the photoresist. Okay, so you have some areas which are now exposed to the metal, and then you have some areas that are covered by this alkyl silane. Now you deposit your aminosilane, so you end up with two self-assembled monolayers next to one another. Then remember at the end of class last time we talked about crosslinkers. Glutaraldehyde is one of those molecules that attaches to amine groups. So the glutaraldehyde molecule, it attaches an amine group here, it attaches an amine group here. So it attaches amine groups on both sides. One end of the glutaraldehyde attaches into these aminosilane areas selectively. Okay, so they self-assemble here. And then you put the protein that you're interested in. Remember, proteins also have amine groups on them, so they will attach to glutaraldehyde. One end of the glutaraldehyde is attached to the aminosilane self-assembled monolayer. The other end of the glutaraldehyde attaches the protein, which in this case is horseradish peroxidase. It's an enzyme. All right, so the point here is that by having two self-assembled monolayers, that helps prevent, one self-assembled monolayer is preventing the attachment of glutaraldehyde, and the other one is enhancing the attachment of glutaraldehyde, which ultimately attaches the protein. So you end up with a nice contrast of one cell type versus the other. So this was used. This used photolithography, traditional photolithography with traditional photoresists, but then it made self-assembled monolayers. Now, another type of approach is where you use light to pattern the biomolecules directly. So these techniques rely on a photochemical change in the protein itself, which causes it to adhere to the surface. There are a few examples here. One was a photoreactive protein. Excuse me. So you can think about this as a protein photoresist. Now, photoreactive proteins are not, they're not widely available commercially, but it did show that some of these can be done. This is a protein that when it's exposed to ultraviolet light, it sticks to the substrate. So you can pattern the proteins this way. Okay. Another approach is to, what's called photo bleaching of fluorophore. All right. So this is mainly a chemistry technique, but there's also some light patterning involved. So I think let's first look at the chemistry of how this works. You start off with a layer of bovine serum albumin. Okay, BSA is a common protein that usually acts as a blocking protein. It prevents the absorption of cells. It's widely available. Okay, so you put this BSA coating on there. Now, the next step is you put down fluorescein molecules. Okay. Fluorescein is a fluorescent molecule. Okay, so you guys have seen glow-in-the-dark watches, or other, there's other types of fluorescent items, fluorescent molecules. These molecules tend to, if you excite them with a certain color of light, they will glow a different color. For example, with fluorescein, you excite it with blue light. That excites the fluorescence, but the actual color that it gives off is green. If you excite a fluorophore with too much light, if you give it too much light, there's an effect called photobleaching that happens. Okay. The photobleaching releases something called free radicals. The free radicals are molecules that have unpaired electrons, which are highly reactive. We can relate free radicals back to when we were talking about photo patterning, photolithography. When we talked about photolithography, we were saying that there are certain chemicals that undergo some type of cross-linking reaction when exposed to light. We were talking about negative photoresists, SU8 for example. The light causes the material to release free radicals, and the free radicals is what cross-links the material. It attaches the polymer chains together, and it changes the properties of the molecule. So this is similar to that sense. Okay, so fluorescein is a fluorescent molecule. It actually, if you give too much light to it, it will generate free radicals. So just like free radicals cause polymer chains to bind to each other in the case of like photoresists, in this case, the free radicals cause the fluorescein to bind to the BSA. So only in areas which are exposed to light will the BSA be bound to this fluorescein. So you can selectively attach the fluorescein to certain areas. And you can see this example here that they've attached these fluorescent molecules selectively to certain areas of the substrate. And they can pattern at very, very high resolution. Now, if you want to attach cells, we'll get to that later, but if you have a biomolecule, this has something called biotin at the end of it. Biotin reacts very strongly, attaches very strongly to something called streptavidin. If you ask any biologist, what's a good binding chemistry, they'll tell you biotin streptavidin. That's one of the first things that is a very well-known binding that happens in biochemistry. Biotin binds to streptavidin, and then you have another biotinylated linker here at the top that is connected to a peptide, RGD peptides that the cells like to attach to. So the short of the story is wherever you pattern this material with light, those are where the cells will eventually stick to. Since you had an assignment on digital micromirrors, this will be interested to know that this type of approach uses something like a digital micromirror to pattern where the proteins attach and where they don't. So you have a light source here. It goes through a spatial filter. So this could be a digital micromirror device set up in a reflective way so that diode would reflect off the micromirror and then go into the camera port. Or this could be an LCD panel. The LCD panel is like what you have on a TV that allows light to pass through in certain areas but not others. Basically, you're sending patterned light into this port of a microscope. It is focused down into a small area by the microscope objective. So whatever light pattern that you projected here, this picture of Einstein that you can see here, it's shrunk down, and it's that light pattern is shown onto your sample. Okay. Now, if this sample contained this chemistry, you would only pattern proteins in certain regions, determined by the pattern of the light. Nice way that you can do patterning just in a microscope setup, which most biologists have. And the last example here is a selective cross-linking of albumin. This was basically showing that that even a protein like albumin, if you illuminate it with certain wavelengths of light, it will cross-link and attach. It'll attach biomolecules to it. And in this case, they use that for neuronal cell culture to pattern where neurons will attach. Okay, so all these techniques, instead of relying on photoresist, they're relying on direct patterning of biomolecules itself without the use of photoresist. A more straightforward way of printing proteins is through microstamping. This can be a very efficient way of patterning proteins. Remember, microstamping is where you took, well, I guess we don't have a diagram of it here. Oh yeah, we do. A few slides earlier. Yeah, with microstamping, you just coat a PDMS stamp with a certain protein, and then you press it up against the substrate that you want to attach the protein molecules to. All right, and this is an example of the patterns that you can get with this. So an important factor here is the wettability of the stamp versus the surface. Now, the surface should ideally be more attracted to the liquid than the stamp. PDMS works well in this sense because PDMS is hydrophobic. If you put a solution of protein on there, and it's a water-based solution, so the water would rather be on the glass than they would be on the PDMS. So this works quite nicely. So wherever you put the water and the aqueous solution, wherever the PDMS stamp comes in contact with the glass, that pattern will be transferred. Okay, you can get very high efficiencies of transferring. This is like what the procedure might look like. This is a very simple process, really. You can deposit this amount of protein on the stamp. The amount of time you spend in contact with the substrate is just 1 second, and almost 99% of the protein is transferred. This is good for one layer of protein, but it's difficult to print multi-layers in this manner. But you can see here that these are all patterns printed from stamps. Now, this part here just highlights the importance of the surface properties of the stamp. In this case, the stamp was, they functionalized the surface of the stamp to either make it hydrophilic and hydrophobic. Okay, PDMS on its own is hydrophobic, right? But they did this experiment just to show the difference if you treat it with something that's hydrophilic versus something hydrophobic. CF3, this was a self-assembled monolayer that was that's terminated in a fluorine group. This is hydrophobic, and this is terminated in an amine group, so this is hydrophilic. You can see in the case where you have a hydrophobic stamp, the transfer is much, much better. Right? You're transferring from hydrophobic to hydrophilic glass, or hydrophobic stamp to hydrophilic glass. So you get a nice transfer efficiency here. And if you're printing from a hydrophilic stamp to a hydrophilic surface, you don't get as good a transfer.
You mentioned last class there corona discharge on the template, because
Yes.
so if it's hydrophobic, you not necessarily, you might not take up the actual liquid, or is that not an issue?
If yes, that's a good point. So if the surface is very hydrophobic, PDMS on its own is hydrophobic. But I don't know if you may have noticed from lab, even though it's hydrophobic, it'll still take up some water. Right. So
It's enough
even
to take up.
enough to do stamping. Right. Right. But you can make the material if you make it. If the PDMS was very hydrophilic, then the transfer part of it wouldn't happen.
You have to
Thank
have
you.
balance.
You have to have a balance, right? If the PDMS was super hydrophobic, then the stamp wouldn't attract any. You wouldn't be able to pick up any of the solution, anyways.
So the material you were talking about doing a corner discharge on to make it a little bit hydrophilic
Mm-hmm.
is super. It needs that to actually take up. I forget what that was, like metal.
Yeah, you can do corona treatments on PDMS to make it more hydrophilic. So, for example, if you were working with a solution and you were having a lot of trouble getting that solution on the stamp, then you do a little bit of corona treatment to it. So it's sort of like, there's a little bit of empirical play here that you have to do. You just know that what the techniques are. The time where you really use corona treatment, where we use it regularly, is when you want to bond PDMS to another piece of PDMS. The corona treatment makes the surface more active, so it bonds better with another piece of PDMS. Yeah. So the corona treatment is used for two purposes. One is to make the PDMS hydrophilic. The other one is to help PDMS bond.
I have a question.
Yes.
What do you mean the printed from CF3 stamp? I mean, the stamp is made of CF3.
The stamp would be coated with CF3. Yeah. So suppose you made a PDMS stamp, and then you put a coating on that stamp to render it hydrophilic or hydrophobic. Okay. It doesn't have to be a PDMS stamp. It could be any stamp.
That's coat with the CF3 or NH2.
Yeah. The CF3 is a hydrophobic coating, and the NH2 is a amine group, so it has more of a hydrophilic coating. Yeah. These are just to show the effect of the hydrophobicity of the stamp. If you want to fabricate multiple layers of proteins with the stamp, you can combine microstamping together with microfluidic patterning. So in this case, they used a PDMS stamp that was a little bit more complex than the basic stamp we saw a few slides earlier. This one actually has a hole in it. The hole is connected to a microchannel. So through this hole, you could inject some proteins and deposit some proteins down in this region. At the same time, you could ink proteins onto this surface of the stamp, and that way you could ink a different protein through microstamping. So you're combining microfluidic patterning with microstamping, and you can get some of these more complex patterns. All right, you can see that they actually pattern 3 different protein types right next to one another this way. All right. Now this type of thing is very interesting from a biologist's standpoint. From what I said earlier, I pointed out in the first slide for today, a lot of times biologists are interested in the interactions between different cell types. So if you can pattern 2 different cell types very close to one another, they'll actually behave differently than if you pattern them very far apart. There's some communication that happens between the cells. Next point here, microstamping proteins with agarose. Jeremy, this brings up the point that you were talking about. With PDMS is very hydrophobic. Will it pick up the inking solution to begin with? So if you use a stamp that is hydrophilic, then it will always pick up. It will always pick up the solution. However, you may have more difficulty in transferring that pattern onto a surface. Okay, But when you're printing proteins with agarose, one of the interesting things about that is agarose is more like a hydrogel. It's more like a sponge, because it soaks up liquid. So if you imagine, like, stamping something with a sponge, you're going to be able to stamp many times because the sponge is going to be full of that liquid, and the capillary action is always going to bring that liquid to the surface of the sponge. So even when you stamp one, it'll get refilled on its own, more or less. So agarose is is a hydrophilic hydrogel. It acts like a sponge. So once it's soaked, it can ink hundreds of times without re-inking, and you can get about 2 micron resolution. Agarose is a material that's it's derived from seaweed, and it's a very, very inexpensive material to get. Biologists use agarose all the time to do DNA separations, electrophoresis. For those of you who've worked with electrophoresis before, the simplest type of electrophoresis you can do is just you buy some agarose. You can use a microwave to make the gel, and you can do electrophoresis that way. So I just wanted to make the point that agarose is widely available, which is one of the reasons why it was used in this application. Okay, same idea of microstamping, except you're using a sponge-like stamp. We already talked about microfluidic patterning of proteins. This slide just goes a little bit into some more details. The idea here was that you had a microfluidic channel, and in that channel you flow the protein solution, and the proteins will physisorb to the surface below it, and then you could just take the channel off afterwards. And this is a very easy and versatile way to print proteins on the surface here. So this was a picture from that famous science paper where they patterned, they created one protein pattern here, another protein pattern here, by having 2 microfluidic channels here on the substrate. So just a little bit of the details here. When you're using microfluidic patterning, one thing you have to be careful about is that you get to maintain uniform concentration of proteins on the surface. Okay, if you use very shallow channels and you rely on capillarity to fill the channels, meaning like you don't have a pump, you just let the channels fill on their own, then what will happen is that the proteins will be depleted later on. You'll have a high concentration of proteins deposited on one side, but fewer proteins deposited on the other side. By contrast, if you have a channel that is continuously perfused, you have a pump going in there, so there's a fixed flow rate going through the channel for a longer period of time, then you get even patterning of proteins. So these are some examples of that. So where they're trying to pattern a self-assembled monolayer using microfluidic patterning. So the only advantage of doing these really small channels filled by capillarity is that you can get smaller feature sizes. 1 micron feature sizes here, whereas with the deeper channels you get a 10 micrometer resolution. Still quite small. Okay. So these types of approaches would be used if you're trying to pattern proteins on a substrate. One of the reasons you might want to do that is to do something called an immunoassay. So this is an application of protein patterning. In a basic analysis of, let's say, a blood serum, for example, just giving a practical perspective. In a blood serum, something you might do at your doctor's office is say, like, well, how much of this protein do I have in my blood? How much albumin do I have? How much creatinine do I have? How much of, how much of what is my protein content profile? Right? The way that proteins are often detected is by something called an immunoassay. So they'll have an antigen. The antigen binds the protein that you're interested in, and they can look at how much protein was bound. Really quickly, let's say your protein looks something like this. We talked about the protein structure. Antibodies can attach proteins. So this is called, let's say this is called an antibody. So antibodies are very specific to certain proteins. So you can have an antibody that will only attach to, let's say, albumin. Or it will only attach to horseradish peroxidase or some other enzyme. So this protein will come and attach to here. If this antibody is immobilized on the surface, well, then now you've basically captured the protein. Now, again, we'll talk a little bit more about ELISA later in the semester, but one thing you can do is after you've captured the antibody to the surface, you can bring in a second antibody, which is called a labeling antibody, and this labeling antibody will either have a fluorescent tag on it, something that will glow a certain color, or it will have an enzyme that creates a fluorescent color. All right, so this is a two-step assay. It's called an ELISA. It's an enzyme-linked sandwich assay. So the reason I brought that up is that you may be interested in flowing in a sample and looking at how it binds with multiple antigens. So one antigen might be specific to protein A, another antigen might be specific to protein B, and so on. So you're detecting multiple proteins in your blood instead of just one. So that's what this mosaic immunoassay can be used for. So what they do is they flow in different antigens, or antibodies, into different portions of the channel. Okay, so you have antigen 1 here, antigen 2 here. Then they add BSA to the rest of it. Whichever parts were not covered by the antigens will then be covered with BSA. BSA is sort of a blocking protein, things to prevent attachment to it, prevent attachment of proteins to the surface in those areas. And then you flow in your sample here like this. Sample 1, sample 2, sample 3. Now, so sample 1 will pass through this region, and then it'll pass through this region. So as it passes through, if any of the proteins attach to antigen 2, this will change the color here. If any of the proteins attach to antigen 1, it'll change the color here. So you can run multiple samples in parallel, and then you get a sort of a graph that looks like this that shows you how much interaction occurred between the different samples, and in each one of the samples you can measure the different antigen levels, or the different protein levels that corresponds to each one of these antigens. Does that make sense? A lot of chemistry. So you'll get a reading that looks something like this. So you'll get different affinities of the different antigens. So this is where you take protein patterning and you actually turn it into a useful diagnostic device. Another way that you can deposit proteins is by particle lithography. Now this avoids the use of photomasks and uses particles instead. I just wanted to note that this is first developed at Wayne State in 2002 by someone in the Department of Chemistry. So remember in photolithography that you have a photoresist layer that acts as a mask. It covers some regions and it doesn't cover other regions. So this was the example that we saw there. Right? Photoresist micropattern. The parts that were uncovered gets treated with a self-assembled monolayer. Then we remove that protein, that photoresist pattern after it. We pattern a second self-assembled monolayer on top of it. So photoresist was used in this patterning step here. In particle lithography, instead of using photoresist, you use a bed of packed beads. Beads are like little spheres that you can buy from a vendor, and those spheres are very narrow size distribution. So if you say like I want 30 micrometer spheres, microspheres, you can buy them from a few different companies, and they will be 30 microns plus or minus 5%. Spheres, if you at the microscale, interactions between the spheres, hydrophobic interactions, will cause the spheres to actually pack together. These spheres pack together into a pattern that looks like this. Now, if you look at this pattern from above, you can obviously see that, like, if you have spheres that are packed together, there's little spaces in between the spheres, right? So this is looking at it from the side view. You'd end up with these certain regions, little spaces between the spheres that are open, that are exposed. Okay, and then some regions of the sphere are where the sphere is in contact with the substrate. Those regions are covered. So you could use particles as a photomask in a sense. Not a photomask, but you could use it as a mask. What they did here is after they put the bead suspension down, they grafted a polyethylene glycol layer. This is a layer that resists the absorption of proteins. Right, so these areas that you see here where the peg only went down in regions which were not being occupied by the particles. Right, so when you remove the beads, you're left with a pattern of a polyethylene glycol, and then when you incubate it with the protein, there are certain regions which are covered with polyethylene glycol, and in those regions the proteins will not adsorb. Only the regions where the protein found the substrate, where there's no peg, those are the regions where the protein will adsorb. The particle lithography is nice in the sense that it doesn't require any traditional photolithography. It doesn't require a cleaner. So a lot of biologists don't have access to a cleaner, so finding ways around that is useful. All right, so let's get into cell micropatterning now. Actually, I think it's if we just take a quick break, I think it's a good breaking point. Let's just take a 2-minute breather, and then we'll continue on to this. I'm just going to go grab a drink of water. Okay. So we talked a little bit about patterning proteins, and now we can talk more about patterning cells. It's going to be divided into sort of two categories here: non-biomolecular templates and then biomolecular templates. These are, more like, just general organic molecules, and these are specifically biomolecules that are used to attach proteins. Some of the concepts you've seen already, but it's just putting the concepts together. So we want to pattern cells, like you saw in the example. A lot of times we want to organize cells on a substrate. Sometimes it may involve one cell type, sometimes it may involve multiple cell types. So how can we do things like that? Among the non-biomolecular approaches, this was interesting because it was, I wasn't aware of this, but the first BioMEMS paper was technically published in the 1960s, where they patterned cells just by using a stencil here. So they basically created a pattern of palladium islands. Okay, so those are the light regions here. And the dark regions here is acetate. It turns out the palladium islands allows cells to adhere, and the acetate regions are non-adhesive. So the cells would basically attach only to the palladium regions. It's interesting that the example that was later shown in the late 90s or early 2000s, there was a famous paper published in Science, was actually similar in some sense to this paper back from the 60s. Sometimes in science, there's certain things people do and then people forget about it for a while, and then that topic kind of like comes roaring back later. It's kind of like fashion trends, actually. Anyway, so this is a slide that shows you, it's just these were things you saw earlier, but just to remind you that self-assembled monolayers can be used to pattern cells. Some of the self-assembled monolayer patterns we saw earlier. We start off with the right here. This was the first example of a self-assembled monolayer that, what happened there? This is the first example of a self-assembled monolayer. The two types of self-assembled monolayers patterned right next to one another. In this example, there was EDA-P, which is a diamine, and then you had a trimine. These two types of molecules had a structure that's similar to something, an amino acid that's found in the extracellular matrix. So these regions here attached the cells, whereas the regions that were terminated in the monoamine versions, or there was no amine at all, those regions did not attach cells. You could pattern cells this way. We saw this example earlier. Then today we talked about how you could pattern cells by patterning the adhesive protein. So if you can pattern an adhesive protein like glutaraldehyde, then just by plating the cells over it, the cells will selectively attach to these glutaraldehyde regions. Okay, so the lesson here is that by patterning the surface properties, you can get the cells to attach to certain regions. Okay, so you don't have to go around and pipette cells and put a cell here and put a cell here. You can plate the cells everywhere, but the cells will only selectively attach themselves to certain regions based on the surface chemistry. This example was based on a hydrophobicity contrast. In these cases, the endothelial cells adhered to hydrophobic regions rather than hydrophilic regions. How did they create this hydrophobicity contrast? Well, they used these polymer micropatterns, and it was a polymer that could be irradiated with light, and that would change their properties. So it could be photopatterned. Okay. So you had the photoreactive copolymers that contain these phenylazide groups. These are the phenylazide groups. The phenyl is that the ring, and the fact that you have the nitrogen there makes it an azide group. They form reactive nitrogen groups when irradiated with ultraviolet light, and this makes the polymer stick to the substrate. So when you cast the polymer, the areas that you expose with ultraviolet light will stick to the substrate. Okay, so this is a mask here. So you're irradiating these areas of the polymer, and so these areas stick to the substrate. The rest can be washed off. So in that sense, it's somewhat like a photoresist. By patterning this polymer, so some regions are hydrophilic and some regions are hydrophobic. They patterned a dimethyl acrylamide copolymer using this technique on a hydrophobic substrate. So they had a hydrophilic polymer on a hydrophobic substrate. This is one way to create a hydrophilicity contrast. The other thing you could do is to have a hydrophobic polymer on a hydrophilic substrate. So example of a hydrophobic substrate is polystyrene. That's, most cell culture dishes, the plastic dishes that you see biologists use, most of those things are made of polystyrene. Very common material. This is generally hydrophobic. Example of a hydrophilic material is PVA, polyvinyl alcohol. All right, so basically you're creating polymer patterns on these things that creates a hydrophobicity contrast. And the cells adhere to the hydrophobic regions. All right, so these were non-biomolecular templates. Now let's talk about biomolecular templates. These are, I would say, more common because these are the techniques that biologists are most familiar with and most comfortable with. So we'll go over several of these examples here. So one way to do this is by patterning physisorption repellent surfaces. So one such example is bovine serum albumin. So bovine serum albumin will prevent the attachments of other proteins and cells. If you have a layer of BSA there, then other proteins and cells will tend not to attach there. Proteins are they're complex molecules. They physisorb to surfaces. They can also chemisorb to surfaces. We've talked about different ways that we can enhance the attachment of proteins to surfaces. There are ways to block them too. BSA is one example. The reason why it's used is because it's quite inexpensive, it's plentiful, and it also doesn't contain RGD sequences. RGD is that peptide sequence that's needed for cells to attach to it. Right? So BSA happens to not contain any of those sequences. So if you coat the surface of a polystyrene plate with BSA, then no cells will attach to it. In this example, they patterned the BSA by using a microfluidic stencil. All right, I'll go over this example later, but you can see what happens is that the cells will only grow in the regions where there's polystyrene, not the regions that are coated by BSA. Besides BSA, another popular non-adhesive surface is polyethylene glycol. So some biomedical implants use PEG. Some forms of PEG are FDA approved, so it is one of those materials that's well known. PEG is a hydrophilic polymer, and this region of the polymer, call it polyethylene glycol because this unit is repeated many times. In this case, it's N equals 70. The length of the molecule does confer some of its non-adhesive properties to it. If you coat something with PEG, proteins and cells will not attach to it. It's a very good repellent background. So I mentioned that in some implants you want to avoid proteins and cells from attaching. So, but you have to take the PEG itself. The PEG itself has to be attached to the surface. All right. So this is an example of where you start off with PEG that's been functionalized with SiCl3. So there was a SiCl3 on this end of it, but that was then attached to the glass. Okay. This SiCl3 chemistry is similar to the self-assembled monolayer chemistry that we saw earlier. Let's just jump up to that real quick. If we look here, there's two kind of self-assembled monolayers. This end, the blue end, is the end that attached to the surface. The thiol group, this SH group, was very good at attaching to metals. The SiCl3 group was very good at attaching to glass. So coming down here, you have this PEG group. This PEG group had an SiCl3, so this is very good at attaching itself to glass. So this is how you get this PEG molecule to attach to the surface of the glass. Now, this particular method allowed you to grow PEG layers on the glass. So the longer the reaction time happened, the longer, the thicker the layer was. Besides PEG, you have other non-adhesives like fluorocarbon polymers. Fluorocarbon is like Teflon. Teflon is quite resistant to cell attachment. There's things like PVA, agarose, and cellulose acetate, so on. So it depends what type of what type of properties you exactly would like, but BSA and PEG tend to be two quite popular protein and cell repellent surfaces. So we have these two chemistries. How do you then pattern them? Okay, we would like to have, like in a culture, we would like to have some areas where the cells attach and some areas where they don't. So how do we pattern these physisorption repellent layers? This shows some examples of that. This is a famous paper, so I think it's worth it to spend some time on this one. This was a PEG thiol group. Okay. We talked about the self-assembled, the thiol-based self-assembled monolayers earlier. So this one is attached to a PEG group. Okay. In the ECM protein, fibronectin binds to the methyl-terminated SAM, but not the hexaethylene thiol SAM. Okay. Hexaethylene glycol, HEG, it has similar properties to polyethylene glycol. It prevents cell attachment. So the cells will adhere to the ECM areas. They do not invade the areas that have the HEG polymer. Let's look more closely at this. This is the fabrication process. You start off with a master and you create a PDMS stamp. So that's parts A through A, B here. With the PDMS stamp, you microstamp a pattern to the surface of, this is a gold layer. Okay, so we're doing a self-assembled monolayer on gold. In some regions, you pattern the self-assembled monolayer that's terminated, that's methyl terminated. Okay, so you're stamping the methyl terminated self-assembled monolayer. You can see the formula here, CH3. All right. This is the region that will ultimately attach the cells. Okay. The regions that are not covered, when we take this whole thing and we dip it into a solution containing the second self-assembled monolayer, the other monolayer will attach itself here in the regions that are exposed. So you get two self-assembled monolayers right next to one another. The cells only attach to the methyl terminated regions, so you can pattern your cells this way. And you can get very fine patterning resolution. This was the example where I have another slide on it later where they showed that the size of the island determines whether the cell survives or not. If the size of the island is too small, the cell will kind of attach itself there, but it's not big enough for the cell to spread out. And it needs to spread out in order to survive. Okay, there's messaging that happens within the cytoskeleton. Remember, the cytoskeleton has these focal adhesion points that help the cell spread out. And if the cell is not able to spread out properly, it sends messages to the internals of the cell to undergo apoptosis, to kill itself. Right? So the environment that you create for the cells is closely linked to the physiology and the biology of the cell itself. Another example here is the interpenetrating networks. We didn't spend too much time on that, so let's not spend too much time on this part of it either. But you can see that you can pattern the cell regions very nicely this way. A little bit more on the PEG thiol cell patterning, because this method has been used quite a bit in many papers. The concept was this: that ECM proteins, the ECM proteins such as fibronectin, vitronectin, and laminin, they attach selectively to methyl-terminated self-assembled monolayers, but not the polyethylene glycol-terminated ones. Okay, like you see here, they only attached to these regions. So you'd create the self-assembled monolayer first, and then treat it with the ECM protein fibronectin. The fibronectin will only attach to these regions, and then the cell comes and attaches to the fibronectin. All right, so the idea is to pattern two self-assembled monolayers, one which absorbs protein, another one which does not. And so you soak it in fibronectin, and then you soak it with the cell solution, and the fibronectin and the cells will go to wherever you want it to go. The cells can survive in these cases for approximately 5 to 7 days. So these were examples of different cell patterns that they did. This is a famous paper published in Science in 2010. It's called Geometric Control of Cell Life and Death. This is what I was talking about at the end of class last time. What they showed was that they patterned these different islands using micropatterning techniques. This was the first time this kind of experiment was done. Now I will say that, remember, it bears a striking resemblance to this experiment in the 1960s, right? But the 1960s experiment could not go down to a really small size. And the reason why this paper was so interesting is that they showed that the size of the island is linked to the cell's biology. Geometric control of cell life and death.
So the paper in the 1960s, nothing died because it couldn't go that small?
Yes, that's right. That's right. They showed in this paper that this was the interesting graph, is that they showed that the size of the adhesive island area, the larger the area, the more DNA synthesis you had. Okay, the way they quantify DNA synthesis is that they fluorescently labeled the nuclei. So they attach like a fluorescent molecule that only goes and attaches to DNA. So the more DNA you have, the more sharply the cell will glow. So it's an optical characterization. The larger the size of the island, the more DNA synthesis you get, and the less apoptosis you get. So the way they measured apoptosis is by labeling the nuclei. You could do something called a live dead assay. It's a well-known assay that will cause the cell to fluoresce a certain color if it's alive. It'll cause the cell to fluoresce a different color if it's dead. There are many ways to look at whether a cell is alive or dead. I don't quite remember the specific technique they used in this paper, but suffice to say that as the adhesive island area becomes smaller, the rate of apoptosis goes way up. And that seems to happen around this region here. So the smallest ones, like once you get around 10 microns and 5 microns, the cell is not able to spread out as much. So this is called mechanotransduction. A mechanical signal is transduced into a signal within the cell. Okay. The size of the island is mechanical because remember last time we were talking about that model that the cell has a cytoskeleton. Cytoskeleton consists of a series of rods, and those rods are under tension. Well, if you're on a really small island, that messes up all the tension between the rods, and the cell is forced up into a little ball instead. The cell cannot survive that way. So it sends a signal to itself to kill itself, basically. They showed us, similar experiments here with what happens when a cell that's on an array of posts. This is pretty interesting. They instead of growing the cells on a flat surface, they had an array of posts, and they found that the cells would actually spread themselves across the array of posts, and they could measure the contractile forces by looking at how much these posts bent back and forth. And this is quite interesting.
Do they vary gap distance? Or?
Yeah, I believe they did. So in this experiment, they had 10 micron gap between them, but there are other folks who are working on like smaller gap sizes. And so when the cell contracts, when the cell's like sort of like feeling its way around, since these posts are so tiny and so thin, the posts themselves will bend a little bit. So you can actually measure the contractile forces of the cell as it's sort of like slithering around the substrate. It's pretty interesting. I mentioned that you can pattern polyethylene glycol layers with stencils. So you just have some the stencil approach that we saw last time, where you have basically a membrane layer with some holes in it, and so this a layer can be placed through those layers, and you can add fibronectin to it. The cells will only attach to the regions that have the fibronectin attached to it. So after you seed the cells, and then you remove the unattached cells, you'll be left with a cell pattern like this. Okay. Now this is a relatively simple technique to do, which is very nice. So you can create these islands of cell colonies. Now this was interesting because they could see cell migration this way. So these red areas is BSA. So those are the regions where the cell was not patterned, and the green areas contained polystyrene, which contained the patterned regions. The cells are labeled blue. So after 5 days, you can see that the cells migrate out of these regions. Now, BSA is non-adhesive to cells. So what do you think happened here? How could the cells actually get out of those non-adhesive regions? I mentioned it very, very briefly last time in class. If the cells have to attach to some type of surface, some type of a surface that resembles the extracellular matrix. So what happens if you don't have that? They generate, yeah. They have ways of actually secreting their own ECM-like molecules. So even if you have a blocking surface like BSA, cells could still secrete their own attachment-like molecules and sort of feel the way around that way.
So if the whole surface was coated, like on a implantable device, do they find that the cells can create stick to that, or do they need like a starting point?
That's a really good question. The question was, if you have an implantable device and you want it to be very non-adhesive to cells, like do the cells need a starting point? Do the cells need a starting point in order to secrete their own ECM molecules and attach to it? It turns out with things like PEG, PEG is very good at blocking. I would say it's better than BSA. So polyethylene glycol also blocks a lot of proteins. So even if the cells are secreting certain types of proteins, those proteins will not get absorbed by the polyethylene glycol, so the cells won't be able to attach to it. BSA might be a little bit different in the chemistry, that if the cells secrete certain types of protein molecules, then yeah, it's not quite as resistant as PEG is. But yeah, that's a good question. A few more examples here. I mean, we're just running the gamut of like different, lots of different examples here. I want to run through some of these and get through them by the end of the class. So I'll go through them relatively quickly, just so you get an idea. Injection molded agarose. So agarose is a material that I mentioned is very popular and very inexpensive. It's known to deter protein physisorption and cell attachment. So you can inject molten agarose into PDMS microchannels to create islands of agarose. So you have a channel here, you inject agarose into it, the cells will only attach to the regions where there's no agarose. So you can pattern cells that way. You can pattern something called a PVA hydrogel, polyvinyl alcohol. That can be assembled on a gold attached self-assembled monolayer. The process really quickly is you have a PET substrate. This is just a plastic, a PET is just kind of like transparency. The same transparency films, that's a PET. Thin plastic films. You can deposit gold on here and create a self-assembled monolayer of the thiolated PVA. And then you coat the PVA on top of here. You do this air drying and rinsing, and this PVA hydrogel, it resists the absorption of cells. So the cells will attach to these regions not covered by the hydrogel. So what these two techniques have in common is that these are materials, these are hydrogels that repel cells that prevent cells from attaching. Now, this is patterning repellent materials. You could do the opposite too. You could micropattern adhesive particles or adhesive surfaces. And this is an example of where you do that. So what are examples of adhesive molecules? Cells adhere to collagen. Collagen is a material that has properties similar to the extracellular matrix. So cells like to stick to collagen, just like they like to stick to fibronectin. Certain proteins you can use to get cells to stick on there. By contrast, BSA, which we just talked about, that's something that prevents cells from attaching. So this example uses both. Excuse me. Let's zoom in here. Now, this example was actually done, I mentioned Sangeeta Bhadia's group, who is doing a lot of co-culture, where they're trying to assemble two different cell types on a substrate. And they're doing hepatocytes, which is like liver cells, for understanding how the liver works. So this is how the process works here. Start off with a glass wafer. You deposit photoresist on here, develop the photoresist, and you have some openings here. So you deposit collagen in these regions. So at this point, after you remove the photoresist, some of the wafer, it has collagen on top of it. When you soak this whole thing in albumin, the regions that were not coated with collagen are now coated with albumin. So you have a contrast, an attachment contrast. The cells like to attach to the collagen areas. They don't like to attach to the albumin areas. So after you seed a bunch of cells and remove the unattached cells, you have a pattern of hepatocytes. Then, now what you do is, okay, you have this albumin-covered region. However, albumin can be overcome. Like if you add a lot of cell serum to this, for example, cell serum has a lot of proteins in it which makes it material attractive to a cell. The cells will stick on there. So if you add the second group of cells and you add serum with it, then now you have the second cell type coating everything that the first cell type did not. So in this case, they had hepatocytes and fibroblasts co-cultured right next to each other. And so what they were doing here is trying to mimic some of the structures in the liver. So this is based on cell substrate adhesiveness. Next example is the patterning of poly-D-lysine. This can be patterned using photoresist. Poly-D-lysine is similar to RGD, that amino acid sequence that's known to promote cell attachment. And poly-D-lysine can be patterned by liftoff and sonication. So remember, liftoff is a process where you first deposit the photoresist. Okay, deposit the photoresist first, then deposit the material, in this case poly-D-lysine, that physiosorbs to the surface, and then you remove the photoresist at the end. So every part that wasn't covered by the photoresist now is covered with the poly-D-lysine. So you have this poly-D-lysine pattern, then you can grow, the cells will attach to just those regions. So you can see some of these techniques involve some photolithography, some of them involve stencils. So there's all these different ways that we can pattern cells simply by patterning the adhesive or non-adhesive molecules on the surface. This is another example of where stencils are used. So in this case, you have your stencil sitting over the surface which you want to pattern. You seed the cells. Okay, you let the cells attach to it. Step 3, and then you remove the stencil, and you're just left with a simple pattern like this. So this was a pattern of cells on a polystyrene dish. Okay, this is very similar to what we saw before. It's just so you can see that the stencil is the thing that's being used to pattern the cells. Okay, so I won't mention any more about that. This is an example of microfluidic and patterning, and here's an example of something new, inkjet patterning. The microfluidic patterning we've seen before, so I won't spend too much time on that. You inject protein solution through the channel. The protein forms a template here, and then you add the cells on top of it, and the cells attach to these regions that were microfluidically patterned. So on this side, this involves inkjets. So we all know about inkjet printers. They're readily available. They're inexpensive. So someone thought that why not use an inkjet printer to pattern where you're going to deposit your cells. Now, one of the challenges with inkjets is that there's two types of inkjets. There's a piezoelectric inkjet, and then there's a thermal inkjet. Most of the inkjets that you have at your home, like the cheap kinds, the inexpensive kinds, are thermal inkjets. You heat up a liquid and it blows a bubble, and that bubble is ejected. The ink is ejected into the substrate, into the paper. Okay. The heat from thermal inkjets are not good for cells. It kills the cells, so you can't really pattern cells that way. Piezoelectric inkjets, by contrast, you can use piezoelectric inkjets to pattern the cells. You have to be a little bit careful because when the cells go through this small orifice of the inkjet print head, there's a little nozzle at the end. If you're pushing a solution through a nozzle at a very high velocity, then it creates a lot of shear forces. And if a cell is going through there, the shear forces can tear up the cell. So you have to be careful about that. Nonetheless, people have printed cells directly using inkjet, piezoelectric inkjet printers. Okay, I don't have too many slides on that, but I do want to let you know that it's possible. There's even a company that sells a printer, a cell printer. So it's pretty neat. I believe they're called BioBot. This example here is where you're using an inkjet printer to print the adhesive protein instead of the cells directly. So you have the ink coming through here and the ink droplets here coming down here, and whenever the way that the inkjet printer works is that you have the print head is going across the surface of the paper. It's raster scanning. You've seen the head going back and forth like this. Together with that, there's a waveform that goes into the piezo crystal. Whenever you apply a voltage to this waveform, this piezo crystal causes a deformation in the membrane here, and that pushes out just one droplet of the ink. Okay. And when the printer says it's like 600 dpi, that means it can do 600 dots per square inch. So the droplets that are coming out are quite small. And every dot that you want to make on the paper just requires one voltage pulse. So you could print adhesive proteins using this technique, and then when you deposit the cells on top of it, the cells will form a pattern. The cells will adhere to those regions. The difference between microfluidic patterning and inkjet patterning is that with microfluidic channels you can get a good resolution, 10 microns or better. With inkjets you can only get maybe 65 microns, typically even more than that. The advantage with inkjets obviously is that it's a very inexpensive process. Buying an inkjet printer might cost if you buy an inexpensive piezoelectric one, it might cost like $1,000 or so, maybe up to $5,000. The cost of a clean room for a lot of the lithography-based techniques is, a clean room might cost millions of dollars to make. So inkjet printing is a nice alternative way to pattern materials. I wonder if we'll get through this. I think we may leave a little bit for next time. This example here is patterns of specific peptide sequences. I want to stress again that biotin, streptavidin chemistry. That's the thing I told you that all biologists will the attachment chemistry that most biologists are familiar with is this biotin, streptavidin. This is based on that technique. So biotin avidin is popularly used in biology. So you can start off with a surface that's presenting biotin groups. This is typically a repellent surface. When you incubate this in an aqueous solution of avidin, these avidin molecules will attach to the biotin. This avidin has a very strong binding to biotin. And then you wash away the excess, and you incubate this with biotinylated ligand here. Okay, and this ligand is one that contains that RGD sequence that cells will attach to. So, I mean, you know, from looking at all these examples, you can start to see that these techniques all have a lot in common. You know, at the end of the day, you're just trying to pattern some adhesive molecules that the cells will selectively attach to. You know, like whether you're doing it by biotin streptavidin binding, whether you're doing it through self-assembled monolayers, you know, whether you're doing it by, photopatterned proteins and whatnot. At the end of the day, you're just looking at a way to pattern the adhesiveness of the substrate to cells. I wish we had time to finish up the rest of these slides today, but I don't want to keep you too much longer. So let's just, we'll wrap up this module on Monday of next week, and then we'll get into the section on microfluidics. Okay? So I'll end there today. All right. So just remember that last time we were talking about these different reasons why we would want to modify a surface. So we talked about like changing the surface properties from hydrophilic to hydrophobic. And there's some examples here. In some cases, you want to treat the surfaces of the microchannel to capture the cells of interest or capture the biomolecules of interest, as you can see in these three examples. In other cases, we want to coat the surface to prevent the adhesion of biomolecules or cells. Okay, so we're just going to finish up talking about methods for patterning cells today. Okay, so among these methods, we talked about ways that you can pattern cells on non-biomolecular templates. So different types of self-assembled monolayers and polymers that resemble the extracellular matrix or the proteins on the extracellular matrix that cells like to adhere to. And then we talked about some examples of biomolecular targets. So these are actual biomolecules that are part of the extracellular matrix, or other molecules that biologists often use to either enhance the attachment of cells on a surface or prevent the adhesion of cells on a surface. So a couple of the examples that we saw. BSA is a blocking protein that strongly binds to surfaces, but it doesn't contain RGD sequences. So it's okay at preventing cell adhesion. It's not perfect. As you can see in this example, after a few days, cells can actually migrate out into the BSA. Now, one of the reasons why this happened, if you recall, that cells sometimes secrete their own ECM-like proteins, and they can secrete their own stuff, or they can go adhere to that. Another non-adhesive surface was polyethylene glycol, popular surface used in a lot of biomedical implants. This is very good at repelling proteins. So we talked about ways that you can pattern a background, physisorption repellent background. So a thiol self-assembled monolayer that was attached to a gold surface that had a PEG group on the end prevented the adhesion of cells. And then you could have other areas, methyl-terminated self-assembled monolayer, which is hydrophobic, and that helps cells adhere to it. So you could pattern where the cells went that way. These are some examples of that. And then we talked about some of the interesting biology that you could do with this stuff. The fact that cells that are on islands that are smaller than the cell, if the cell is not able to spread out properly, the cell will basically commit suicide. It will undergo apoptosis. These are some other cell repellent materials. Agarose is known to repel cells, and it's very cheap, widely available. That's one material you can use. PVA hydrogel is also a cell repellent surface. We talked about that. There are ways that you can create co-cultures of cells. If you can create structures similar to what's in the body by co-culturing two cell types. So you can do two layers of cell patterning. In this case, the first layer, the first patterning, which was done through a stencil, was used to pattern photoresist stencil was used to pattern one layer of cells. Okay, and then the second layer was placed after that. So this was used to make structures that resemble what is in the liver. Remember that cells often behave differently when they're in vivo, when they're in structures that resemble what's inside the body, than they are when they're just grown on a petri dish. Okay, these are other examples of that. We started talking about microfluidic and inkjet patterning of ECM proteins. So there is also a way that you can pattern cells directly. You can 3D print cell structures now. However, one of the issues with that sometimes is that the thermal inkjet printers, they generate a lot of heat at the nozzle, so this can kill certain types of cells. Piezoelectric inkjets, which basically have a structure like this. A piezoelectric material is a material that expands when you apply a voltage to it, and it does so in a relatively predictable manner. Higher-end printers tend to have this piezoelectric inkjet technology. So as the print head is scanning over the surface of your paper, there's also these voltage signals that are being placed on the print head, and that injects the ink from the nozzle at predictable times, deterministic times. So the size, the volume of ink that's ejected by the print is typically on the order of picoliters. Very, very small amounts of ink that's basically determined by the duration of the voltage waveform. Okay, so this is an example of an inkjet printhead. Now, piezoelectric printheads don't have the problem of high temperature, but when the cell is going through a very small orifice like this, they can be exposed to high shear rates, high shear stresses. And those shear stresses can actually tear up the cell as it's going through the nozzle. So again, we have to be careful when you're trying to directly print cells on a substrate. That being said, there are direct cell printers that are available right now commercially. So what this slide is talking about is the way that you can pattern ECM proteins instead. Pattern the adhesive protein and let the cells attach to them on their own. So these were a few examples of that. This was a microfluidic patterning where you're flowing the ECM protein solution and letting that attach to the substrate, and the cells will eventually attach to that. And you can also use inkjet patterning to deposit these protein layers, and the cells will then attach to that. The difference in the two methods, that microfluidic channels give you a very small resolution, less than 10 micrometers, while the inkjets are typically 65 microns or larger. The cheaper inkjets, are probably looking at 100 to 200 microns. High-end inkjets can get maybe 65 or a little bit less than that. Okay. So the obvious benefit of doing something with an inkjet printer is that it's maskless, right? You just give you can give it a file to print, telling where you want to print the cells, and immediately you get your patterned output. So this is really useful for prototyping. Okay, you guys saw in the lab like the way that PDMS devices are made is first you have to go and make the mask. You wait for your mask to come in. That takes a few days. And then you have to do a lithographic step to make the mold. That takes a few hours. Then from the mold you make your microfluidic chip, and then if you can use your microfluidic chip to then pattern the proteins. So there's a long turnaround time, and in research we often want a very fast turnaround so you can iterate your designs very rapidly. So in those cases, the maskless approaches are often better suited. And for the biologist who doesn't really care that much about microfabrication techniques or how it's done, they care about what is done. They just want to pattern the cells on a surface in a certain pattern, and then their focus is really on to study the biology of it. Okay, so techniques that are more or less off the shelf and are I wouldn't say brainless, but they're turnkey solutions. Those are often the types of solutions that a biologist will prefer to have. So we talked about this a little bit the other day. I'll just go over it again. The biotin-avidin binding is a popular a chemistry that the biologists like to use. So this example was how that biotin-avidin binding could be used to pattern cells on a surface. So you begin with a surface that has biotin groups, but it's mounted on polyethylene glycol, these PEG. Now PEG is it's a long chain molecule, so that's why it's represented by a string here. Now these PEG chains on their own will prevent the attachment of cells. If you incubate this whole setup in a solution of avidin, the avidin binds very strongly to the biotin. Okay. Once you allow the avidin to bind, you wash away the excess, and then you incubate with a biotinylated ligand. So you can see the interaction is happening between the biotin and the avidin in both cases. So this is sort of like a sandwich type approach. The avidin is really just being used as a glue to attach two of the biotin molecules together. Now the second biotinylated molecule has ligands attached to it, and those ligands enhance the attachment of cells. They may contain an RGD sequence or this other sequence that's known to attach certain cell types. So we're going over a lot of different chemistries. Different chemistries are more or less comfortable to different groups of biologists. Some biologists really prefer to have the biotin-avidin binding. Others are more comfortable with self-assembled monolayers. Some are comfortable with polyethylene glycol. And some are very comfortable with just polystyrene, the petri dishes that they've been using for long periods of time, and collagen. Okay, so there are many methods out there to be able to pattern cells on a surface. This slide talks about microstamping cells. So in this case, agarose stamps are being used to pattern bacterial cells as well as mammalian cells. We mentioned agarose stamps in last lecture and a few lectures earlier. Agarose is a hydrogel. So it's sort of a porous hydrophilic material which soaks up water. Soaks up water like a sponge. These hydrogels can be used to pattern these types of cells because when they absorb the liquid, they bring the cells to the surface of the stamp. The liquids can go into the pores of the hydrogel, but the cells, because they're larger, they can't go through the pores. So the cells are basically on the surface here. Okay, so this is an example here. You have an agarose stamp with positive features on it. Let's say, so. Okay, positive features means features sticking out of it. When you add the suspension of cells, because the hydrogel is hydrophilic, it's going to absorb that solution. So the cells will remain on the surface here. When you invert it and print it, the bacterial colony will be printed on the petri dish like that. And you can make relatively small micropatterns this way. Okay, these are other examples of patterning bacterial colonies. You can see that in some cases you can get very, very tiny colonies and get you can make large-scale arrays of these types of colonies for high throughput studies. The last point here is that the cells actually don't attach to the stamp. Okay, they're on the surface, but they don't attach because agarose is a cell repellent surface. So it's very easy to transfer the cells to the media or the whichever surface you have, if that surface is more adherent to the cells than the agarose is. So this is yet another way to do microstamping of cells. More examples of direct inkjet printing. I mentioned that you can do direct inkjet printing, but again, you have the issues of the temperature. So this is an example of a modified commercial thermal inkjet printer, and in this particular example, they used Chinese hamster ovary cells, which are relatively larger cells. They're also more robust than say neurons, which are more flaky cells. The cells sometimes can survive the inkjet process, but not always. So the high shear stress and the high temperature are sometimes a concern there. A few hundred microns is the resolution that you can get with direct inkjet patterning. Now, this is two-dimensional inkjets, but you can extend this to three-dimensional patterns for tissue engineering. Tissue engineering is a field that is growing rapidly, and the field of tissue engineering attempts to build tissue outside the body using cells from a donor. So let's say one of my organs is damaged. They could take cells from my body and potentially grow me another organ. That's the vision of this field. Obviously, it's not there yet, but in order to grow certain types of tissue, for example, vasculature, if you want to build a blood vessel outside the body, you may take endothelial cells, but if you just put the endothelial cells on a petri dish, they will grow out into one flat surface. They're not going to create a blood vessel. What tissue engineers often do, and there are there are entire semesters worth of classes in tissue engineering, is that they create a scaffold. Okay, they have to put the cells out in a certain pattern that resembles vasculature. Then the cells on their own will actually form these types of structures. Okay, but they need a little bit of guidance at the beginning. It's like when you grow a plant and you have rods holding up certain parts of the plant in order to get the plant to grow. Cells actually do communicate with one another, but and the way they communicate with each other actually helps determine what the ultimate structure is. But the way that tissue engineers often do this is they create a scaffold that mimics the structure that they're trying to create in the body, and then they seed the cells onto that scaffold. Okay, and in these types of approaches, often they use three-dimensional printing techniques to print out the scaffold, a three-dimensional scaffold structure that might resemble a certain organ, and then they might seed the cells directly onto that scaffold. And in other cases, they can directly 3D print the scaffold as well as the cells with it. Okay. Obviously, we're not going to be spending much time talking about tissue engineering because that's another topic on its own. But it's a very interesting area that uses microfabrication. I mean, you can look at these examples that we saw earlier. This is an example of tissue engineering in a two-dimensional space. You're putting two cell types right next to each other. And in fact, one of the groups that does this, like Geeta Bhatia's group at MIT, they're one of the leaders in leading groups in tissue engineering. They do two-dimensional techniques as well as three-dimensional techniques. But this is just a small example of what is done in that field. I don't know if any of you heard recently. Last year there was an article published about they were able to create a beating heart in a petri dish. Anybody hear of that? If you take cardiac myocytes and you grow them in a dish or in a structure that resembles that helps the cells form like this kind of like cardiac tissue, then they will start to beat on their own. Pretty neat. Another approach to single cell patterning is to actually use flow in a microchannel. Okay, instead of like just seeding out the cells on a plate and letting the cells attach and then washing the excess away, which is what we've been basically talking about the last, this entire lecture. Another completely different approach is to use, microfluidic channels, specifically like cell traps. This was published last year, not last week, because last year I was making up some of the new slides. The idea here is to trap single cells in a microfluidic device and then transfer those trapped cells to a surface. So let's zoom in here. This is the concept. First of all, the first thing you have to do is is to trap single cells in a microfluidic device. So let's say you have a channel that looks like this, okay, and in that channel you have this structure that looks sort of like an L-shaped structure. When there is, and by the way, next to this L-shaped structure there is a small gap here. All right, so when there's no particles or cells over in this region, the flow lines look something like this. Okay, we're going to be talking about laminar flows and microfluidic flows in the next module, so it's a good lead into that. So there's essentially two paths for a particle or a cell in the flow to follow. If the cell happens to be in one of these streamlines, it'll just flow right past it. If it happens to be in this blue streamline, it will get stuck right here. It'll get caught here. Okay. The interesting thing is once a cell is trapped here, notice that this trap is just large enough for a single cell. When the cell is trapped here, this flow gets blocked. As a result, this blue streamline disappears because no flow can come that way. Instead, what happens is that the flow lines go back this way. So the remainder of the cells will actually go right past it. So in that sense, the trap has a feedback mechanism. Once a cell is trapped there, only other cells will literally just go past it. So you get a single cell trap. It's a very simple and elegant approach to doing single cell trapping. And as we'll talk about later, one of the things that microfluidics is really good for is to be able to trap individual cells and do various types of experimentation with them. So the dimensions here are quite small. You have a 3 micron gap here. The size of this little trap is about 12 microns, the size of a typical cell, and the dimensions here about 22 microns. Okay, so you can see examples of where single cells have been trapped here. Now what you do is you can take, if this was the microfluidic chip that you made, this was the upper part, this is that PDMS layer, and this brown region is just the material underneath. It could just be a flat sheet of PDMS. It could also be a glass cover slip. This upper layer is used to trap the cells, okay, and then they just remove the upper layer, and then they have their adherent cells here. Okay. Now, there's a little bit more details to that. They actually had to treat this surface here in order to get the cells to adhere to it. Okay, but we've already talked about many ways that we can enhance the adhesion of cells on a substrate. Okay, but the interesting thing here is that the channel itself, which was used for patterning, can just be removed at the end. Okay, so the reason why this was published in this journal, PNAS, Proceedings of National Academy of Sciences, it's one of the top journals in the world. Science, Nature, and PNAS are like the 3 really big journals in all areas of science. The reason why it was published in that journal is because you get very high resolution, but you also get very high cell viability. Okay, the ability to trap just single cells and to create patterns of single cells, it's very challenging. You can create colonies of cells, groups of cells, but patterning single cells on a surface is a little bit more difficult. Also, this technique it got the single cell array, but also had very high cell viability, and it had a relatively rapid turnaround time, about 30 minutes. A lot of these incubation protocols where you're incubating the cells and growing the cells on a surface, you have to wait several hours for the cells to attach, and then you wash them away. So this is a very interesting technique. You can see they made large arrays of single cells, and then they transferred them to a surface. This green fluorescent dye shows that they are viable. So what they do is there's a fluorescent dye that they can allow the cells to uptake, and that will cause them to fluoresce green when they are stimulated with blue light. So the cells fluoresce in this diagram, and it also indicates that they are alive. These are more examples of different patterns they did. This is just to show that you can pattern them in different shapes depending on how your microfluidic channel is designed. And this final example showed that you can pattern different cell types. Again, there's a lot of interest in understanding interactions between individual cells. So this is a way to do that. Still another approach is using a scanning probe. How many of you have any of you worked with atomic force microscopes before? I think I asked the class this, the first lecture. Yeah, when we were talking about AFM probes. Okay, we do have a couple AFM probes in our engineering college. I think Dr. Mao in chemical engineering has one, and Dr. Cheng in our department has one. I talked about atomic force microscopes earlier, so I won't mention too much about that. The scanning probes, the idea behind scanning probes is that you have a very sharp tip that you raster scan across the surface. You drag it over the surface in various patterns. Now, in this case, the scanning tip is a microelectrode, which is used to electrochemically remove BSA, albumin, in the regions where it's scanned. So remember that BSA is a blocking protein. It prevents the adhesion of cells in those areas. So if we remove BSA from certain areas, the cell is more likely to adhere to those regions. So how do we remove it? In this case, they're using an oxidizing agent, which is generated electrochemically by the electrode. So electrochemical probes use a voltage. They're running a voltage between the electrode and a reference electrode, which is out in the solution somewhere. The solution is being held at a specific voltage by the reference electrode, and then the microelectrode is at a certain potential. Okay, for those of you who remember a little bit of electrochemistry, if you put a voltage on the electrode, that voltage can initiate a chemical reaction under certain circumstances. So in this case, the microelectrode is generating an oxidizing agent which removes the BSA from the albumin from certain areas of the chip, and that the cells are able to grow on those oxidized regions. Okay. Again, this is a maskless approach, so you don't need to have the lithography, and you can basically just by scanning your microelectrode over there, you can get these different cell patterns. And another example here is the electroactive patterning of self-assembled monolayers. The self-assembled monolayers we talked about earlier, okay, they attach to the surface through a bond between the sulfur and the metal atoms. But it turns out that those self-assembled monolayers can be removed by applying a voltage to the metal. Okay, applying a voltage to the electrode generates charge on the electrode, and presumably that charge can take the self-assembled monolayer off. So it's electrochemistry. I Obviously, I'm not going into all the details of the electrochemistry, but the basic thing here is that it's an electrical desorption. The cell repellent regions can desorb if a pulse is applied to the gold layer underneath it. Remember, the several monolayers are being attached to a gold substrate. So wherever you remove, whenever you remove, this is an example of the cells were patterned, and these regions out here were cell repellent, and then a voltage pulse was applied to remove the repellent layer, and after some time, after 600 minutes, you can see that the cells invade into those regions. Okay, so this is kind of interesting because you're using electrical control to determine where the cells adhere and where they don't adhere. Okay, so many different chemistries that are used to pattern surface properties, proteins, and ultimately cells for various types of biological assays. There's a lot of concepts here, but a few take-home concepts. So there's a few popular chemistries and general principles that are used for attachment. Besides the ones I've shown you in this module, besides the ones that are shown in the textbook and other resources, there's tons of different attachment chemistries. All you have to do is go to Sigma Aldrich or one of these other websites, Thermo Fisher, Pierce, like any of these websites, they have like hundreds of different attachment chemistries. Okay, obviously we can't go over, there's no point in going over all of them. If you know some of the basic concepts, then the rest is just shopping, really. And in fact, that's what we do. A biologist, if they're trying to attach the cell to a certain type of substrate, they'll try to find the correct antibody that will bind to a certain cell type that they're trying to capture. But it's helpful that you know that these attachment methods exist and where to go to find them. So a few of the popular chemistries: self-assembled monolayers attach to metals, if they're a thiol self-assembled monolayer, or they can attach to glass, in the case of silane-based self-assembled monolayers. The end groups can be functionalized to enhance or prevent the absorption of proteins in cells. Remember, the self-assembled monolayers have two sides, the side that attaches to the surface, and then the end group. So something like the methyl group, CH3, can enhance the absorption of proteins or cells. By contrast, polyethylene glycol, which is a long chain hydrophilic polymer, that can prevent the absorption of proteins in cells. Self-assembled monolayers can be used as, with crosslinkers. The crosslinkers are middlemen, so to speak, which attach the self-assembled monolayers to proteins. We talked about glutaraldehyde, NHS esters that bind amine groups. Remember, proteins have a lot of amine groups on them. Okay, so they're good at, these two chemicals are good at binding the amines. The malamido attaches to thiol groups. If you don't have attachment chemistries, proteins will still adsorb to many surfaces through many different types of physisorption mechanisms, ionic, hydrophobic interactions. Okay, many different mechanisms that we talked about. So there's a lot of reasons why proteins will adsorb to the surface. Oftentimes we have to prevent the adsorption of proteins to the surface. They often attach by default. So the ways that we can prevent the adsorption is by things like polyethylene glycol, BSA, interpenetrating layers. We didn't talk about IPNs too much, but PEG and BSA are pretty common. Cells, if you want to attach cells, they like to attach to proteins in the extracellular matrix or peptides which resemble sequences in the ECM. So some cells will secrete their own attachment proteins if none are available, so they can invade into regions that don't have attachment proteins. To prevent invasion, efficient patterning techniques should have both an attachment region and a protein repellent region. Okay, you notice that a lot of the techniques we talked about earlier, they did two patterning steps. One was for a protein enhancing, absorption enhancing region, and one was a repellent region. That gives you better contrast between for the cells to specifically adhere to the attachment regions. And polymers with high hydrophilicity or hydrophobicity contrast can also attach cells. So you can do a lot just based on hydrophilicity and hydrophobicity. Okay, these self-assembled monolayers are, in some ways they're dependent on hydrophobicity. This methyl group here is hydrophobic. PEG is hydrophilic. Okay, so polymers with high hydrophobicity and hydrophilicity can also create a contrast for cell attachments. All right, so this is all chemistry. These are the attachment methods. Now, for the engineer, they're often interested in how can we pattern those attachment methods. How can we say, I want it to attach here and not here? So we talked about photo patterning. Photolithography we already know about. We talked about in the previous module. We talked about how to photo cross-link certain types of these attachment chemistries. You can photo cross-link certain polymers. You can photo cross-link some of the middlemen, so to speak. You can use light, ultraviolet light, to remove self-assembled monolayers after they've been put on. So that allows you to pattern SAMs. Another method was microstamping with PDMS or hydrogels. Then microfluidic patterning is where you put a channel on top of it and you flow liquid through the channel and then remove the channel. That was used to pattern proteins. We saw that earlier, and just now on the last slide we saw that you can also pattern cells using that technique. Stencils are very straightforward, nice thing to use. They're just thin membranes of the PDMS or some other material. You put that down on the substrate. In some areas the fluid goes in the other areas they don't. And then inkjet printing, which can be done in 2 dimensions. It could also be done in 3 dimensions. All right, so that's it for the this patterning module. Do you have any questions about any of the stuff we've covered so far? Okay. And I'll just end with this, that depending on the cell type that you're trying to pattern, or the biomolecule the type you're trying to pattern, every different situation is going to have a certain biological question you're trying to answer. It's going to have certain cost constraints. It's going to have certain materials which are allowed or forbidden. You're going to have to, you may have a certain specificity that you require. How much of, if you're trying to attach one cell type, how much of another cell type can you tolerate? So all of these specifications go around in selecting which type of attachment chemistries or surface treatments you're going to do on your devices. Okay, so we'll just end there. I think what we could do now is to take a short break. Let's take a 5-minute break, and then we'll go on to the next microfluidics module.